Thank you, Dr. Szelski. Our last was sort of be aspirational or future-looking. Dr. Jobin.
Thank you. I'm not texting. I'm just trying to link my presentation with the phone, so the right side of the room could see a laser so that everyone will be happy, I guess. I was sitting there. I was having a hard time following. So thank you for the organizing committee for inviting me. It's kind of a little bit brave because this field of microbiome and cancer is kind of nascent and exploratory and frontier. So I don't have answer to you and to this is the new marker you have to follow. It's just to put the seed in your brain and to pay attention. And I think somehow everyone is paying attention to this field now. Right, so we're not alone. There is a lot of microorganisms, and they are intrinsically, I need to click this, click that, here, and yeah. So they are intrinsically linked to anything that, physiology-wise, that we face. And some research have been linking different diseases to microbial composition. So if you look at the stools of patients, you will be able to distinguish case and control looking at the stools and the composition of microbes. So that put the field very up front of therapeutics and diagnostic. And some of these diseases are, cancer related. So that was also interesting for the cancer field. So should we open a new front in the fight against cancer, including the microbiome as a new entry point? There's microbes everywhere in our body, surface cavity, but the hub, the headquarter, 95% of the bugs live in the gut. So they will be located, most of them in the colon, but ileum, the small bowel have 10 to the 12 microorganisms. It's a lot of people, and they are there for a reason. So they're rich and diverse. And how do you study them? Most of the time, you can't culture these things, very difficult for analysis, so you do genomic approach, and you've been familiar, the cancer field has been familiar with genomics, so there's nothing fancy by that. It's just the pipeline is a bit different, so you have an ecosystem, so it could be, tissue, biopsies from cancer, feces from patients, and so on. So you have your sampling, and then you isolate nucleic acid, DNA, RNA, depending what you want to do. And then you do your, depending on the material of the material you isolated, either you do what we say a 16S ribosomal amplification, PCR base for taxonomy, whole genome, metagenome, the entire genes of the ecosystem, metatranscriptome if you isolated RNA, and then you feed that to, you sequence. Once you sequence, you get a bunch of information and you process them. So this pipeline, I think, I've been using different biological contexts. So the microbiome is not very different. But there is a lot of caveat to this pipeline, computational-wise, which kind of pipeline are you using, which kind of technique you've used to isolate the genetic material will have a huge impact on the output, what you're going to measure. So there's a lot of, throughout, there's different pipelines. So a group will like to use pipeline X, Y, Z, and it's not the same as you. So how much comparable the data are across studies, that's an open question. But I have to mention that this is probably the most impactful step into microbiome analysis. So if you have been following this field a little bit, there were a couple of papers. One that was quite early. It's about drug toxicity and microbiome. That was when I was at UNC, led by a chemist, and I thought that he was speaking a different language than me because I was using microbiome in a different way than toxicity. So, but that paper kind of was not cited that much, and then pick up pace later on when people realized, I think, that there's something into that. But there's other paper in 2012, 2013, that plant the seed that microbes may be important in this is all animal studies, so that microbiome composition, and specifically a couple of species, could be very important in teaming up with different drugs. And I have something here that, I wrote a little piece of that because I thought it was quite interesting. So in a nutshell, you could split this into different things. So toxicity or efficacy. And the toxicity is because microbes has a big, large set of genes, way more than us, and they have genes that could metabolize different drugs that impact either lower toxicity at different site, or increase the metabolism to increase efficacy, or team up with different drugs to increase different aspect of the immune response. I'm not here to tell you, explain you in much detail. The goal here is to say there is microbes that could interact with drugs, either toxicity, efficacy, and they are going through different processes, immune response or pure metabolism. Okay. And there were papers that were published a couple of months ago in Science, 3 papers, and that was quite interesting, and I think really set the field in motion. These papers were from France, Gustave Rossi, this is Chicago, and this one was from MD Anderson. And what they did, they look at patients, I will come back to that, which kind of cancer, it's mostly melanomas or lung cancer, and they look at the feces of these patients, and look for, it was a cross-sectional, some were doing a longitudinal, but all of them did cross-sectional, and they look at the signature, and what they could find is that a responder and non-responder were different in their microbiome. Some were driven by increased Bifidobacterium, Akkermansia, Faeculobacterium, and non-responder may have more of these bacterial EDs. So that was a signal that was interesting on its own. But what they did is to take the feces of these patients, responder and non-responder, transplant that, or fecal transplant them into a mouse, a preclinical model where they have implanted tumors, and do the PD-1, this was a PD-1 study, and look for cancer response. And you could see that the microbiome implanted from a responder patient were having a better reaction in the mouse as well. And then they could, draw a conclusion into which immune cells were infiltrated in the tumors and how do you link this phenotype to the microbiome. Needless to say that was something that attracted a lot of attention. Again, this is very early on in the game. There's like a handful, maybe 10 papers about the entire microbiome and immune response. So we're not here to tell you which markers. But the thing about that, though, is that this is PD-1, patient treated, and everyone receives the same drug, and yet there were a lot of difference in the signal. One group said this bug, the other group said that bug, and one said, no, that bug. So if you were into biomarkers, it's a bit confusing. Which one are you going to use? So, the beauty of sequencing is that the data are public. So you could download them if you have space, and every cancer center has a lot of space, so you download them, and you look at the signal, how these guys did that. Could I reproduce the signal on my own when I analyze that? So this is what we did. This is not published, but that was just for this meeting. I thought it would be interesting for you. So why will you be spending time looking at microbes in patients? I mean, like I said, you could generate biomarkers of responsiveness. We could identify a signal that could be milk into a therapeutic response. If you do synthetic cocktail of microorganism, you could come and compensate for a poor signal in a patient in the hope that it will link the immune system better to your immunotherapeutic intervention. So that's the thought about that. Why am I going backward? It should be, oh no, okay, no, we're good. So this is the question, why 3 different signals? Why don't we have one cluster of microorganisms that we could use? So this is a breakdown of what they did, okay, so it's different groups. So V4 is a segment of the ribosomal, so the Wargo or MD Anderson and the Chicago group looks and did pretty much the same disease, the same sequencing technology. They did metagenome, and they did 16S and the T0. The French group did something a little bit different, no 16S, that's whole genome, metagenome. Iron proton is a different technology. Lung cancer mostly, and longitudinal, so pre and post treatment. But if you look at these 2 groups here, you could cross-reference these. Why don't they have the same bugs, right? So that was something that was interesting to us. So we went to download all these data, and there's a lot of data, especially the home genome, metagenome, and reproduce every figures. And the good news is we were able to reproduce every figures. We were able to reproduce that each group have their own signal. So it's true that they find 2 different bugs linked to responders. So that was good. And this is just an example. Okay, so we did that. This is our own data. They didn't do a PCA. And the thing that they were doing very unconventional analysis of microbiome. So we apply a very simple pipeline that many people could do without any filtering, without any extra step, and we're able to generate a principal component analysis, discriminating non-responder to responder. Every dot is a microbiome of one patient. Okay, so responder and non-responder has a different microbiome, and who is contributing to the signal that says that they are different. And if you do, this is a ELF6 analysis, so you could see there's different clustering of bugs at hierarchy. So you're interested in getting species level because that's the most precise definition. And the species level for the group from MD Anderson, we found Faecalibacterium protnetzi, which the same bugs that they found. So everything checked in terms of what they did. The bad news is none survive FDR. So if you have a large data set and you apply FDR, which you should, everything disappeared. There's nothing. So how do you move forward if you don't know that your signal is statistically relevant? So that's a question that I leave to you, but in the field of microbiome, we like to put FDR. So, and then we do the whole genome, metagenome on all these things and studying, reproducing what they found. So there's no difference, responder, non-responder, across the study. Where is that thing disappeared? I don't know. Okay, so all these different studies, there's no clustering whatsoever. Okay, so that was one. And what we found is the clustering, this is the same, different color code, but this is the same plot, color for different things. Here's different, is color by studies, which means that this is an American study, this is an American study, this is a French study, and they cluster according to population. So, the Ruti drives some sort of difference because they are French patients. And so there's a difference in microbiome composition, but there's no clustering across. You combine every data. You would like to have a bigger view, put more data, and do you see a signal using whole genome metagenome, which is more higher resolution than the 16S gene amplification, and we see no separation whatsoever. So that was something, but if you have access to data, could you milk the data to guide you into generating biomarker, yes or no, coming from the microbiome field? So that will be very important. Am I flashing yellow? Yeah, I'm good. So then we wanted to see if we could do that, and why did we thought it was a good idea? This is whole genome metagenome. This is not for immune checkpoint, but this is a study that was published 2 years ago, and that was for disease. Okay, so they downloaded exactly like we did. They downloaded a whole genome metagenome that people publish in different disease and use machine learning. Machine learning is like a subset of artificial intelligence. So you ask the machine to process a lot of data and find me a profile, find me some sort of a direction into these data. Do they point to something? So in an untrained fashion. So here is a ROC curve. You could see that cirrhosis, if you look at the microbiome, it's a very high predictor of cirrhosis. Then you have colorectal cancer, IBD, and less of obesity and type 2 diabetes. So that was done before. So there's no reason to think that you could not do it in a population that had been exposed to different drugs, cancer drugs, especially immune checkpoint. When we did machine learning on that, different machine, right? So lasso, random forest, and SVM, and then also, so nothing showed up. No predictor were found with machine learning. And then if you shuffle label, you even fool a machine, because if you have a signal, you shuffle label, make sure that your signal will survive that. Actually, it performs a little bit better. So everything is random. So the bad news is that people would think that the 16S, if you send a granule microbiome, you say, I'm going to do 16S, people will start being a little bit angry at you. You say, I'm going to do metagenome because I capture more information. I just gave you metagenome here. You cannot make it. This is metagenome on taxa. So you assemble the metagenome to find taxa, which is better resolution than the 16S. So what to do? Oh, the machine is blocked here. So the idea here, in terms of what I've been presenting to you, is that The conclusion are from these study or reanalysis of the studies. The difference reported in tax are not pipeline dependent. They're related to and not related to the choice of bioinformatics. So that's the good news. So you could reproduce data. The results are reproducible as an independent pipeline without the need to filtering steps. There's no shared signal across the 3 data set. Each has its own signal. And applying machine learning to predict our responder and non-responder didn't succeed. So I have to give up because this is wireless, and the wireless gave up on me. So no fancy stuff anymore. So we have data, though, that if we assemble the metagenome in function, then you could see responder, non-responder. So you have to go beyond the typical taxonomy. You have to go into function, gene function, assemble the microbiome into a gene function pathways, make machine learning, find pathways, and try to validate these pathways if you're in the function. So I think there is hope for that. So in terms of cancer microbiota, I think that there's a lot of research and interest into the human genes, obviously for good reason, and this will be the distribution of microbial genes. I think there is a case for we should incorporate them into our equation and understand the interaction between these microbial genes and function with anything that is a cancer process. And if you look at this is my last slide. This is a Salvatore Dali. It's a picture that it's swam on the lake, and you see these swans, beautiful. And there's a promotion aspect of microbiome in cancer. It's been well studied. And you look at the reflection here. The reflection is elephant, very different than the swan, which could be your therapeutic. So depending how you look at the microbiome, you could see the progression, the promotion of cancer. You could also harvest and understand the microbiome in terms of therapeutic interaction. Okay, so that was a lab meeting. I showed up at that meeting. They dress up all like me. It freaked me out. But Rad Garibay was the guy who did all the informatics, a fantastic person. There's a lot of work into understanding this process here. And I think this meeting is showing that there's interest in bringing the microbiome. There's a lot of companies, startup or established companies are going after that. And I think it's important to take it into context. So thank you for your attention.